Assalamu alaikum, dear students. Welcome to the course Principles in Animal Life 1. This is lecture number 34. The topic is carbohydrates, polysaccharides. In our previous lecture, we discussed the first two types of carbohydrates, and now we will discuss the third type. Discussing agenda for the topic will be introduction, polysaccharides and its types. Lecture outcome: after watching and listening this lecture, students will be able to know about polysaccharides and their various types. Introduction. Polysaccharides. Polysaccharides are macromolecules and they are polymers with a few hundred to a few thousand monosaccharides joined by glycosidic linkage. So the polysaccharide comprises of few hundred to few thousand monomers, the basic building blocks or the simple sugars, and these simple sugars are linked via glycosidic bond, and the reaction which links them is the dehydration reaction or the condensation reaction, in which the water molecule is removed. Some polysaccharides serve as storage material. They are hydrolyzed or broken down into monosaccharides when they are needed. For example, in plants, the surplus energy is stored in the form of starch. In animals, the surplus energy is stored in the form of glycogen. And when the animal is fasting, when the animal is in need of energy, and the animal is starving, no food is available, then those reserves of energy are broken down into their individual monomers, monosaccharides, and then energy can be yielded from them. Remember, in our previous lecture, we discussed that an individual cannot derive energy from a disaccharide. First, the two monomers must be separated from one another. So similar is the case with the polysaccharides. The polysaccharides must be broken down into their individual monomers so that they can be utilized for the energy. Other polysaccharides serve as building material for structures that protect the cell or the whole organism. The structural polysaccharide of plants is cellulose. The structural polysaccharide of animals is chitin. The architecture and function of a polysaccharide are determined by its monosaccharides and by the position of its glycosidic linkages. So what they want to say is that what function will be performed by a polysaccharide is determined by the fact that what is the composition of the monomers and what type of linkages in which linkage the monomers in that polysaccharide are linked with one another. So the nature of the monosaccharides and the type of association between them will determine the function of that polysaccharide. First, we move on to the storage polysaccharides. Both plants and animals store sugars for later use in the form of storage polysaccharides. When the energy is in surplus amount, then it is stored. So sugars are a source of energy, so we directly refer to the sugars as energy. When it is in surplus amount, they are stored in the form of starch in plants and in the form of glycogen in animals. In animals, the glycogen is primarily stored in the liver, and when the animal is starving, suppose there is a hibernation period, or the animal is unable to find its prey or food for a certain length of time, then those reserves are utilized to run the affairs of the body. Plants store starch, a polymer of glucose monomers, as granules within cellular structures known as plastids. So starch is the storage polysaccharide of plants, and starch is stored in plastids. Plastids include chloroplast too. Synthesizing starch enables the plant to stockpile surplus glucose. So when the plant is synthesizing starch, then this plant is in a position to store the surplus amounts of glucose. Surplus amounts means those which are not required, which are in abundance and can be stored for later use. Because glucose is a major cellular fuel, starch represents stored energy. So because the glucose is stored in the form of starch, glucose is a source of energy, so here the starch is referred to as stored energy. Now here we have the structural illustration of starch and glycogen. Here you see the starch, which is a storage polysaccharide of plants, and it is stored in plastids. And remember that starch is of two types in plants. One type is amylose. Amylose is unbranched. There is no branching in amylose. The other type of starch is amylopectin. Amylopectin is branched, but not excessively branched extensively. It is branched at certain points, but that branching is not extensive. In the case of animals, the branching in glycogen is extensive branching. The sugar can later be withdrawn by the plants from this carbohydrate bank. Carbohydrate bank means starch in case of plants and glycogen in case of animals. And the reaction in which the polymers are broken down into their individual monomers is called hydrolysis, which breaks the bonds between the glucose monomers. Which type of bond? Glycosidic bond, which is a covalent bond. Most animals, including humans, also have enzyme that can hydrolyze plant starch, making glucose available as a nutrient for cells. So this starch is not only a stored energy for the plant itself, but it is also a source of energy for its consumer, for the animals which feed upon those plants. This reserved energy can be utilized by the consumers of the green plants. Now, potato tubers and grains, the fruit of wheat, maize, rice and other grasses are the major sources of starch in the human diet. The starch is stored in wheat, maize, rice and other grasses by the plants for later use, and when the humans consume those green plants, then this reserved food materials becomes part of the animal's body and can be utilized for the derivation of energy. Most of the glucose monomers in starch are joined by 1,4 linkage. 1,4 linkage means the carbon 1 of one monomer forms bond with the carbon 4 of another monomer, like glucose units in maltose, the one we saw in the maltose in the previous lecture. The simplest form of starch is amylose. This is one type of starch and it is unbranched. Amylopectin is another type of starch and it is branched. There are branches emerging which has 1,6 linkage at the branch points, and we will see it in a while in the figure. Now, animals store polysaccharides called glycogen, a polymer of glucose that is like amylopectin but more extensively branched. So the glycogen is like amylopectin, but amylopectin is less extensively branched, whereas there is considerable branching in glycogen. Vertebrates store glycogen mainly in liver and muscle cells. Breakdown of glycogen in these cells release glucose when the demand for energy increases. So the liver is the center of the metabolic activities, and the muscles continuously they move in order to move the animal. So these are the two areas which are in a continuous need of energy, and they are supplied with the reserves which can yield them energy. The extensively branched structure of glycogen fits its function. Now, glycogen is mentioned on the previous slide. The branching is more extensive in glycogen, and because the environment inside the body is aqueous, so this extensive branching helps glycogen to dissolve in water. More free ends are available for breakdown. This stored fuel cannot sustain an animal for long, however. Means although it is a form of energy, a form of stored energy, but the animal can only be the animal's life can be sustained for a short period with the help of the stored carbohydrates. It cannot sustain the animal's life for longer periods. This is signify the fact that the carbohydrates are stored and then they becomes the source of energy, then they are again stored depending upon the availability of food. So you can say it is a short-term storage of energy. In humans, for example, glycogen stores are depleted in about a day unless they are replenished by eating. So if you are observing a fast, then your glycogen reserves will be depleted at the end of the day, and when you take food, then some of the food will be stored back again in the form of glycogen. This is an issue of concern in low carbohydrate diets, which can result in weakness and fatigue. Now, structural carbohydrates. Organisms build strong materials from structural polysaccharides. For example, the polysaccharide called cellulose is a major component of the tough walls that enclose plant cells. Globally, plants produces almost 10 raised to the power 14 kg. That is 100 billion tons of cellulose per year. It is the most abundant organic compound on Earth. Cellulose is the most abundant organic compound on Earth. Like starch, cellulose is a polymer of glucose with 1,4-glycosidic linkages, but the linkages in these two polymers differ. The difference is based on the fact that there are actually two slightly different ring structures of glucose. Recall from our previous lecture when we said that the 5-carbon and the 6-carbon sugars, they adopt a ring form in the aqueous environment under physiological conditions. Physiological conditions are those which are found inside a living system, inside a living organism or a cell. So when these straight chain glucose molecules they adopt a ring form, then when you analyze the hydroxyl group at position 1, the hydroxyl group will either be below the plane or above the plane of the ring. When it is above the plane of the ring, then it is beta glucose. When it is below the plane of the ring, that is alpha glucose. Now remember that in starch all the glucose monomers are in the alpha configuration, means they have their hydroxyl group at position 1 directed downwards below the plane of the ring, whereas in cellulose the hydroxyl group at position 1 is directed above the plane. So in cellulose there are beta glucose monomers, and in starch there are alpha glucose monomers, and 1,4 linkage means that the carbon at the molecules at position 1 of one sugar forms bond with the molecules at position 4 of another sugar, and this is referred to as 1,4 linkage. In the coming slides we will also study 1,6 linkage, which is the point of branching. Now glucose monomers of cellulose are all in the beta configuration. The hydroxyl group at position 1 is directed upwards above the plane, making every glucose monomer upside down with respect to its neighbors. Now the differing glycosidic linkages in starch and glucose give the two molecules distinct dimensional shapes, 3D shapes. Certain starch molecules are largely helical. They are in the form of the helix, and this configuration is efficient, helps in efficient storage of glucose units. On the other hand, a cellulose molecule is straight. Cellulose is never branched, and some hydroxyl groups on its glucose monomers are free to hydrogen bond with the hydroxyls of other cellulose molecules lying parallel to them. So the cellulose molecules are not branched, they are lying together, and then there is interaction between the hydroxyls of both the strands, and that provide strength and resilience to these cellulose molecules, and in turn to the plant. In plant cells, parallel cellulose molecules held together in this way are grouped into units called microfibrils. So this is a microfibril which consists of cellulose molecules, unbranched cellulose molecules held together by hydrogen bonds. These cable-like microfibrils are a strong building material for plants and an important substance for humans because cellulose is the major constituent of paper and the only component of cotton. The unbranched structure of cellulose thus fits its function, imparting strength to parts of the plants. Enzymes that digest starch by hydrolyzing its alpha linkages are unable to hydrolyze the beta linkages of glucose. Now here is another example of the fact that the enzymes have specific substrates where they act. So the enzymes which digest the starch by hydrolyzing its alpha linkages are unable to hydrolyze the beta linkages of cellulose due to the different shapes of these two molecules. Starch is the stored form of energy, so when a plant needs this stored form of energy, then this plant has specific enzymes which degrade the starch component and does not degrade the cellulose component. Why? Because of the differences in the glucose monomers. So the cellulose is not harmed In fact, few organisms possess enzymes that can digest cellulose. And in animals and humans, there is no enzyme which can break down cellulose. So remember, cellulose is not a nutrient for humans. But when cellulose move in the intestinal canal, it provokes the adjoining cells lining the cavity to secrete mucus, and this mucus secretion eases or helps the passage of food materials across the alimentary canal. Remember that cellulose is not a nutrient for the humans, but it is beneficial because cellulose provokes the cells of the alimentary canal to secrete mucus, and once the mucus is released, then it helps in the movement of food material and the undigested food in the gut. Thus, although cellulose is not a nutrient for humans, it is an important part of a healthy diet. Most fruits, vegetables, and whole grains are rich in cellulose. On food packages, insoluble fiber refers mainly to cellulose. Some microorganisms can digest cellulose, breaking it down into glucose monomers. You know about the termites. They have a flagellate protozoan in their gut which breaks the cellulose for them. Similarly, the cow also harbor cellulose digesting prokaryotes and protists in its gut. These microbes hydrolyze the cellulose of hay and grass and convert the cellulose to other compounds that nourish the cow. So here you have so this is starch. Starch is a storage polysaccharide of plants, and we have discussed 1,4 linkage and 1,6 linkage. Now 1,4 linkage is present in the main stem, and 1,6 linkage is present at the branching point where a branch emerges. So of course there will be 1,4 and 1,6 linkage only in those carbohydrates which are branched. The carbohydrates which are not branched, they will only have 1,4 linkage. So starch is branched but less extensively branched, and glycogen is extensively branched. You can see here the branch emerges and the sugar of the main stem and the sugar in the branch are associated through 1,6 linkage. Cellulose is unbranched, whereas The bonding pattern is 1,4 glycosidic linkage. Now we have the structural polysaccharide of animals, which is chitin. Chitin is the carbohydrate used by arthropods to build their exoskeleton. Exoskeleton are hard cases that surrounds the soft part of an animal. Made up of chitin embedded in a layer of proteins, the case is leathery and flexible at first, but becomes hardened when the proteins are chemically linked to each other, as in insects, or encrusted with calcium carbonate. So chitin is also found in fungi, which uses this polysaccharide rather than cellulose as the building material for their cell walls. And chitin is similar to cellulose with beta linkages, except that the glucose monomer of chitin has a nitrogen containing pigment. So this differentiates the glucose and chitin. The similarity is that there is beta linkages. The difference is that the glucose monomer of chitin is attached with nitrogen. This was the end of the lecture. Thank you. Allah Hafiz.